as we were saying, the issue is that power is bounded above by 1, and so this rejection ratio can't get any larger than 20. So even with our optimistic range of our prior of 0.01 probability that there's really an effect from this SNP, our pre-experimental odds for a given SNP can't get higher than 0.2. So we can't think there's more than a 0.2 odds for there really being an effect. Now, if we instead we use the genome-wide significance threshold, which we'll be talking about, this 5 times 10 to the negative 8th, now power divided by our significance threshold is bounded above by 1 divided by 5 times 10 to the negative 8th, or 2 times 10 to the 7th. So we can get much, much stronger evidence from statistical significance with this much more stringent threshold. And what that means is that when you see a hit that reaches genome-wide significance from a study with adequate power that of course also has to deal with population stratification, the other issues, but assuming it's a good study design and it gets genome-wide significant, it's credible even with pessimistic priors. So here's the same kind of calculation we just did, but now assuming the genome-wide significance threshold. Now you can see you have almost no power with samples of even tens of thousands of people. With 100,000 people, you only have 16% power now, which means that of the SNPs that have this 0.02% effect size, we're only going to detect 1 out of every 8 of them. But conditional on having observed them, even with the low end of our prior probability, we can be extremely confident that it's a real effect. Yeah.
Too strict for social science because we're pretty sure our effect sizes are small. Therefore, the probability that a given gene. So like if you were really doing medical stuff where you thought there was only like 1 or 2 genes in the whole genome that did it that had huge effects, that would be one thing. But since we know it's very highly polygenic with all the social science examples we've done, this seems way too strict.
Well, this could be. I think a lot of people feel that genome-wide significance is too strict, but it's. I think that there is a frequentist justification for it, which we'll talk about. It's a Bonferroni kind of correction, and it's become conventional in the genetics literature. And I think that part of why it's become conventional is that there was this history of non-replicable results or inconsistent replications, and so the medical genetics community has decided they don't want to have any more of this problem of replicability. They want solid results they can really believe, and even if that means having a more stringent significance threshold than is necessary, or they want to they just want to have really solid results that they can believe in. Yeah.
I guess the interesting question is relative to if we assume effect sizes of the size that we have in social science, then how much does this now traditional threshold, how much does it increase the sample size that we need to declare victory on something? Is it? If it's only, it might be a factor of 2, it might be a factor of 4, but that's kind of the cost to keeping this now traditional standard.
Yeah, so it's a good question. I don't know the answer. One thing to keep in mind is that I think a lot of so we're talking about discovery of individual SNPs. A lot of the useful results for social science come out of the polygenic scores, which don't impose, the most predictive scores don't have any significance threshold at all on them. So this stops being an issue. So this is really for identifying the SNPs that we're going to then say there's some causal gene somewhere, and we're going to try to learn about its biology. But I take your point. Yeah. So then you make a compelling case about not using
using an ex post frame, or we're not learning from an ex post observation of the effect size.
Yeah.
Do you feel like that insight carries over to the significance threshold as well? So if I observe a result with a high degree of significance, can I make conclusions ex post based on the significance level at which I observe the result?
Yeah, I mean, I think so I think you should, once you've observed the result, take into account the data that you've actually collected, and you would want to condition, if you end up getting a result with a really low p-value.
But what I'm saying is, if I get a result with a really low p-value.
Yeah, it's a more convincing result, and so that's why I was saying these calculations are pre-experimental. They're assuming we don't actually know what the outcome is other than significant or not, so it's really for experimental design purposes. If you know what your outcome is, you should be conditioning on what the outcome was, and then you want to calculate a Bayes factor, which is the way of doing this calculation that incorporates that information. So there's the Bayari et al. Paper on the reading list discusses Bayes factors. Okay, so there's some other important insights that come out of these calculations. One is that, so remember I said on this slide, a hit from a genome-wide significant from a well-powered study is credible. Well, what does well-powered mean? Well, we have this, our normal intuition is 80% power is well-powered. When we have such a stringent significance threshold, you can get a very, very persuasive result with 16% power. Why? Because this rejection ratio, 16% divided by the significance threshold, is 3 million. It's pretty strong evidence. So I think a more general lesson, which I'm going to come back to, is that you don't want to be relying on heuristics when it comes to evaluating evidence. You want to be actually learning how to do these calculations, which is why we're trying to teach them to you on the problem set. And you want to do these calculations because our intuitions often are wrong, and our heuristics are often misleading when taken to some new context. So our heuristics from a lot of usual standard social science research is you need 80% power. Turns out for genetic association research, 16% power is totally fine. And we hear this from reviewers, people who react to our papers without having thought deeply. They say, "Oh, we only had 16% power. It's not a credible result." Well, you go through the calculation. It actually is a very credible result from 16% power. Yeah.
I think I have two questions. So the first one is that like this is sort of like assuming that you're doing essentially a candidate gene test, but like you're using that significance threshold, right?
Yeah.
But really, we're testing 100,000, 1 million candidate genes, so that number would go down a lot, right?
No. So for this kind of Bayesian calculation we're doing, you can do it SNP by SNP. So the relevant information, I mean, Bayes' rule shows you the relevant information is, what your significance threshold was and what your power is and what your priors are.
So it doesn't change whether we do 1 or a million.
That's right.
And then my other question was, so earlier we were talking about how with the Progenic score right now, it's sort of we just take everything in. We don't worry about the significance threshold, and we say like the stuff below it actually has more information than noise in it. So we just kind of like don't have enough sample size. Like when do you see that changing? Like it seems like at a certain point, it's going to be better for prediction to only include the things that like are useful, right?
Yes.
And so are we near that? Like is like educational attainment 3 going to be like more predictive if you don't use everything, or what do you think?
Do we know the answer to that?
We're going to talk about this more on Monday. I mean, in principle, you could do a correction that downweights things that have, so like a standard Bayesian correction, which is going to downweight things that are smaller more than the things that have. But you still want to give, like at least a little bit of weight. You'd have to a little bit of weight to everything. You can show that reduces mean squared error to at least assign a positive weight to everything that you have, as long as you do the weighting correctly.
What you're describing, though, if you do the weighting with just the beta hats, then what you're describing has already happened for a lot of traits, like height, for example.
Okay,
So
like things
if you
that
plot predictive power against p-value thresholds, like an inverted U, where you do well as you progressively relax the p-value threshold, at some point you just start adding noise.
But that's if you're not doing any of this funny Bayesian shrinkage,
Yes,
right?
yes, it's without any of without penetration.
Yeah.
So we're there with height, but we're not there with like these more sort of distal behavioral traits.
Well, we may be there. I don't know. Are we there with education yet?
No, I still think we do progressively better as we relax the p-value threshold. But Robbie would know. Yeah, he's nodding his head.
Yes.
Yeah, okay. All right, so bottom line is do these kinds of calculations. All right, so let's talk now about genome-wide association studies. So what is a genome-wide association study? Well, the medical genetics world went from one extreme, which was having a hypothesis going into the study about each SNP that they're testing, to the other extreme. We can't rely on these hypotheses. We don't really know enough about the genome for them to be all that useful and change our priors all that much. We're going to just go totally atheoretical and just let the data tell us what are the associations we should be following up on and giving us information for future studies. So GWAS is an atheoretical testing of all of the SNPs measured on the chip. So these days about 2.5 million SNPs, or you can impute them up to something like 9 million SNPs, as I think Aisu will probably be talking about some. The standard level of significance is the genome-wide significance threshold. There's a frequentist justification for that threshold, which is, remember we said there's about 1 million independent loci in the genome in European descent populations. So you divide 0.05 divided by 1 million, and that gives you 5 times 10 to the negative 8th. There's also a Bayesian justification, which we just talked about, which is that this is you need to get in this ballpark, maybe not quite this stringent as Miles was saying, but you need to get much lower than 0.05 given that we have a low prior probability in order to get convincing enough evidence. Okay, so then in GWAS we're going to have the same causal model that we've been talking about, the additive model. But we immediately run into a problem when we think about estimating this model, which is the dimensionality problem. So if you try to run this regression in Stata, it's going to not run, give you an error message because you have 2.5 million regressors and 100,000 people, and it's going to say can't do that. So the standard solution is to run a set of univariate regressions where we estimate each beta individually for each of the 2.5 million SNPs. So we do 2.5 million regressions. Now, you might immediately wonder, we have better solutions than that. There's been a lot of work in machine learning and estimating regressions where you have more regressors than observations. So why not do that? Well, to date, there have basically been 2 barriers. One is that in order to get the kind of sample sizes we need, this 100,000 plus samples, until very recently, there was no UK Biobank, there was no 23andMe sample that had hundreds of thousands of people. There were these medical cohorts with a few thousand people that had to be that we had to rely on exclusively, and so these data sets have IRB and privacy restrictions so that they can't share the data, or you can't go in and analyze the data yourself. They have to run the analysis. So we had to do these studies by meta-analysis. We had to ask each cohort to run their own GWAS and upload their results to some centralized server, and we get this beta coefficients and standard errors, and we meta-analyze z-statistics, and we meta-analyze them together to get the final GWAS results. And the machine learning methods are designed to work on individual level data. Okay, so that was one problem. The other problem is even if you could find some way, even if you could find some data sets that were able to pool the individual level data, it's very computationally intensive to do some of these kinds of methods when you have millions of regressors and thousands of people. Yeah, Miles?
I think there's a third thing to say, which is that the things you are doing that you're headed towards are not that different in spirit from idea that machine learning is some magic, I think, is it's in spirit the kind of thing that you end up doing.
Yeah. So one thing so because of these kinds of constraints there's a premium in genetics research on developing methods that can work with GWAS summary statistics, which means the results that come out of the meta-analyses, which are just from these univariate regressions. And there's a lot of methods that end up producing results that are very similar to what you would have gotten if you could have done machine learning on the individual level kind of data. So there's been a lot of creativity in developing those kinds of methods, I think consistent with what you're suggesting. OK, now I think these barriers are actually lifting now. So there's increasingly large samples like the UK Biobank that you can get individual level data on. And there are, Amazon is now offering some options for secure cloud computing. And there is software under development that will allow for genetic analysis on cloud computing. So I believe that within the next few years, we're going to start seeing more sophisticated analysis methods. OK. So, but as of now, we're doing these univariate regressions. So we have this causal model, and this model is actually the univariate model is the one we estimate. So an important consequence of the fact that we're running univariate regressions is that the thing we're estimating, this univariate coefficient, is not the same as this average causal effect that comes out of the model. In particular, the GWAS coefficient that we're estimating is this causal effect, but plus the effects of other SNPs that are in LD with that SNP. Okay? And so as a result, the genome-wide significant SNPs are typically correlated with other SNPs that are in LD with it. And what we actually get out of a GWAS is not very confident evidence that a particular SNP is associated with the phenotype, but rather evidence that some region, a SNP and its neighbors, which are in ILD with it, are associated with the trait. And so we call that region a locus. That's typical terminology in GWAS. And then you often hear the term lead SNP used to refer to the SNP in the genome-wide significant locus that has the smallest p-value. So that's often you just report a single SNP, you report the one with the smallest p-value. Yeah?
So maybe you're into this, but when the winner's curse kind of suggests that it's more likely that it's one of the SNPs in your cluster than necessarily the lead SNP in terms of the lowest p-value?
Yeah. I mean, the one with the smallest p-value has the highest probability of being the one in some kind of purely statistical sense, not taking into account other information.
That are like tightly linked.
Yeah. But yeah, you can't be very confident that that's the one, and there's surely going to be a winner's curse, and that's actually what Patrick is going to talk about in the next lecture. Okay. Now, and this is, ISO, I believe, is going to talk about this more about constructing the set of lead SNPs from a set of GWAS results. But by the way they're constructed, because we're just picking one SNP in each locus, we're constructing a set of approximately uncorrelated SNPs. Okay? So we're dropping all the ones that are the other significant SNPs that were correlated with the lead SNP in the locus. Okay. So how does GWAS address the gene discovery problems? Well, for multiple hypothesis testing, we're going to measure all the SNPs with our, we're going to test all the SNPs in our genome-wide data. And we're going to then adopt the genome-wide significance threshold to Bonferroni correct for all this multiple hypothesis testing. Now, I think that one of the potentially negative consequences of data like the UK Biobank becoming available is that now we can get back into a world where people could do tests with many different phenotypes and not report all of them. So I'm not sure that we have a permanent solution to the multiple hypothesis testing problem, but at least in the to date in GWAS studies, it's such a big effort to run one of these meta-analysis studies and coordinate all these cohorts that people focus on a single trait at a time typically and publish the results that come out of it. Population stratification, let me defer to Patrick's lecture on stratification. He'll talk about how the fact that we have genome-wide data allows us to use strategies like constructing principal components from the data that can control for ancestry that weren't available when we had traditional candidate gene studies and only measured a few variants.
Yeah.
Yeah.
Question.
Yeah.
The last point you're making with the MHT, the danger of phenotypes. I mean, if you have, 1, 2, there's 1, 2, and 3, could you just conformally correct at the, for like 5 times 10 to the -8?
Yes.
Like divided by 3.
Yes, so you could. What I'm worried about is, I guess more technically, it's it's uncorrected for multiple hypothesis testing or publication bias, where people might, run a bunch of different GWAS on the UK Biobank, look at the one where they got some genome-wide significant hits and only publish that one and not correct the p-values or report that they studied other things, or specification searches for a given phenotype. Yeah.
That's where I think this 0.5 times 10 to the -8 being strict is going to take care of you, because as long as the number of phenotypes checked, and I don't just mean in one study, across the whole range of studies, is less than the typical number of genes in a social science that affect the typical social science phenotype, then you still should be in pretty good shape.
Yeah, so it does build in some protection against, builds in kind of some additional Bonferroni correction to the extent that it's too stringent. Okay, so let me skip the stuff on stratification. Okay, so let's talk about how these consortia have dealt with the problem of low power. So I mentioned there was this 2005 landmark paper by the Wellcome Trust Case Control Consortium. After that paper, and with the growing availability of genome-wide data, the medical genetics community shifted almost completely toward doing GWAS studies rather than candidate gene studies. And here was one of the early papers on schizophrenia. There was an early paper on schizophrenia published in 2009. The x-axis, so this is a kind of typical way that you see GWAS results presented. It's called a Manhattan plot. On the x-axis, we lay out all of the SNPs on the genome-wide data. So this region here is chromosome 1, this is chromosome 2, chromosome 3, and so on for all the chromosomes. And then we have each location in the genome where we have a measured SNP. So each of these points represents a SNP and its significance with the association with schizophrenia. The y-axis is the p-value on a negative log 10 scale, which means that higher numbers are more significant. It's lower p-values. This red line corresponds to the genome-wide significance threshold of 5 times 10 to the -8. So dots that get above that threshold are more significant. It's called a Manhattan plot because you kind of see these towers that are like the skyline of Manhattan. Why are there towers?
Multiple correlated SNPs.
Yeah, exactly. It's the LD structure. It's we said that you have a locus where there's a SNP that's a causal SNP that's associated. Its neighbors also tend to be highly associated. So you see these towers in the data. Yeah.
How should we think about the size of the beta coefficients relative? So I know this doesn't tell us anything explicitly about them, but so the sample size is the same for all of these individual tests, right? So
More or less.
Sometimes there's not. Is it like that?
There could be variation it could be that a particular cohort that contributed to the GWAS didn't get a measure of a certain SNP, and then the sample size will be smaller for that SNP. But
They use different SNP chips, or what's the?
Sometimes they use different SNP chips, but sometimes it's just something goes.
You see in one cohort and not the other.
Yeah.
So that, and then also independent of the effect size that's measured, that, and then also I guess just the variation that you see in a SNP, are those the things that are going to affect the p-values? I guess if a SNP like, if there's low variation in the SNP, then we're even.
Yeah. So there's a few things that end up affecting the p-value. One is in expectation, the larger the true effect, the more significant the association is going to be. Another thing is the larger the sample size, the more significant it's going to be. Also, the more frequent the SNP is in the population, the higher the minor allele frequency, the more power, more power you have to detect it. Okay, so this was an early study of schizophrenia. This was actually a big study for the time. They managed to pull together 2,600 schizophrenia cases and about 3,300 controls. This was multiple cohorts contributing to this study, uploading results to a server and meta-analyzing the results. And they ended up finding 0 genome-wide significant sites. So so that was 2009. By 2011, they were able to, this is done work by the Psychiatric Genetics Consortium, they were able to increase their sample size to about 9,000 cases and 12,000 controls. Then they got 5 genome-wide significant hits. They were able to further increase it by recruiting more cohorts. By 2014, they published what's now viewed as a landmark paper, even at the time, was viewed as a landmark paper in Nature, with 37,000 cases, 113,000 controls, with 108 genome-wide significant sites, that then subsequent biological analysis of things along the lines of what Fleur and James were talking about, and Tonu's going to talk more about next week, has led to, they argued, new insights into the biology of schizophrenia. So I want to point out that actually, in my view, GWAS has been an unmitigated success. Okay, so this is some paper by Peter Vischer looking back in 2012 over the first 5 years of GWAS, looking at the number of publications of GWAS studies, number of SNPs discovered, cumulative number of SNPs. If we extended this to today, it would just been exponential growth. There's been many, many discoveries. This is looking at how the number of hits on a log scale has depended on sample size for a number of different traits. Again, if we extended this, the trends would continue. The reason Peter Vischer and co-authors had to write this paper in 2012 is that GWAS has gotten a pretty bad rap in the world of funding and the science community more generally. In the early days, the results were like these results of the 2009 schizophrenia paper. Most of the GWAS were finding nothing, or just a small number of genome-wide significant SNPs. And so the critics said, it doesn't work. You just don't find anything with GWAS studies. Vischer and colleagues wrote this paper to say, look, the problem was small sample size, that the study design does work, and in fact, he also argued that the results were much more replicable than much of the earlier work. Lately, GWAS still has a bad name in funding in medical circles, and the reason now is that they say it's passé. Like, now we can do sequencing. And so we shouldn't be measuring genome-wide data anymore or doing genome-wide association studies. That was, 5 years ago. So if you hear people, saying negative things about GWAS, it's probably because of one of those two perspectives, which are still, I think, very common. Yeah.
So they don't like to fund canon gene studies. They don't like to fund GWASs.
Well, the medical genetics world, they want to fund sequencing, or
They're going to fund me to sequence genomes as a psychologist.
Okay. Well, so I don't know if they'll fund a psychologist because they're real scientists. And I'm caricaturing the kind of no, I mean I think my point here is just that we need to make the case. We always need to be making the case to our biologist colleagues and to funding agencies that GWAS is worth, is worth pursuing because it is actually paying off for the kinds of complex traits we're interested in, and it seems like a productive approach. Yeah.
Now it seems, now obviously it's cool to do full sequencing, but I think it would be easy to exaggerate the benefits of the sequencing because of linkage disequilibrium. It's going to be hard to get a strong signal that that differentiates between this gene and one, 1,000 bases over. And so I guess I'm a little skeptical about the power for distinguishing exactly which gene it is as opposed to a region even with the sequencing data.
Yeah, I agree. I mean, you can do more. You probably can do better with higher resolution genotyping or sequencing. But I basically agree that for most of what we're interested in, at least for now, genome-wide data are plenty good. And they tag almost all of the variation in common single nucleotide variants. And and I think that people, what you'll hear a lot of times is that people will say, well, there's all this missing heritability. There's the, you just found all these SNPs in GWAS, but they don't come anywhere close to what we know is the heritability of the trait. That must mean that the stuff you're measuring, the common SNPs, just don't account for that heritability. It's because of these rare variants that aren't measured on your genome-wide data, and you're going to need sequencing data to get those rare variants. And I think a big part of the answer I mean, that's true. There are rare variants that contribute to some of the heritability, but I think a big part of the answer to why we haven't discovered, we're not explaining as much of the variance with the genome-wide data is because the sample sizes haven't gotten big enough yet. And we'll get, there's a lot more progress to be made just with genome-wide data.
So if it was rare variants that were actually explaining missing heritability, you'd have to be getting effect sizes that were similar, like ApoE and things like that, right? Like it'd have to be huge.
Well, I mean, they're not going to be as strong as the ApoE effects, but they probably are stronger than the effects of common variants. There's reasons to believe that there exist rare variants with stronger effects than common variants. But I mean, I don't want to get into this now, but there are ways to try to estimate how much of the variance is explained by common variants versus rare variants, and I think the general the general result for the kinds of phenotypes we're interested in is that the rare variants explain much less of the variance, and that's for the kind of theoretical reasons that David talked about in his talk. That if you look at the
low frequency, smaller effect size.
With low frequency, yeah. Let me not go into that.
Yeah, and we don't talk about more. Just quickly, what is a rare variant for common variant? Is 1% or less, or like what do you?
It can be defined in different ways, and it's kind of arbitrary. A typical threshold that's used is 1% minor allele frequency.
One way to think about the sequencing data is probably, I'll bet the sequencing data only gives you much mileage statistically if you're doing non-SNP stuff. And so the non-SNP stuff probably is important, and the sequencing data is probably a fair bit more convenient for non-SNP stuff than the
Absolutely, yeah. That's one of the big advantages of getting the sequencing data is to look beyond, to look at structural variants and indels.
Or de novo things.
Or de novo mutations, yeah, which are not likely to be so important, for explaining much of the variance. Sorry?
What does de novo mean?
De novo means that it's new in that generation. So when you get a mutation, it gets passed along to the children because it's in the DNA. If it's in the sperm or egg cell, it gets passed on to the children. But a de novo mutation is one that you get because of exposure to ultraviolet radiation.
If I have a de novo mutation, like what's the likelihood that it's like in all of my body versus just like parts of it?
It depends on when you got it. Like,
yeah.
Just going to add to that, de novo can also occur in the sperm or the egg. So it can be in the germline cells of the parent, but not in the parent's somatic cells. So it can occur at that stage, and then at fertilization, that individual will have that mutation. But if you got the blood from the parent, you wouldn't pick it up. It's only if you got the sperm cells from the parent would you see any of that variation. That's also another.
So
I heard someone estimate once that every individual has about 20 de novo mutations in their own DNA. So they're not. I mean, 20 out of
Yeah.
3 billion. But you know.
I guess it's like someone who doesn't really understand biology. If I have one, say it happens in my fingernail, what are the odds that my de novo mutation ends up in my foot, right?
Yeah, so I think the ones that we're mostly thinking about are the kinds that Fleur are talking about. It's like mutations that are happening in meiosis.
Okay, yeah.
that end up in all of your cells of your body. If they end up in just a particular place later on, it might cause cancer.
Yeah.
But it'll be restricted. Yeah.
So as new GWASs have come out, have they confirmed the results of previous GWASs in the sense that the same loci are?
Yeah, the large-scale GWAS have proven to be very replicable. I'm going to show you an example of that soon. Yeah. Yeah.
I had a question about increasing the sample size. So as you're getting larger and larger sample sizes, you're also getting samples from more diverse environments.
Yep.
You would think. And so, you mentioned earlier that the SNPs that you tend to discover are the ones that are robust across environments. And so one would think that as you expand the sample size and you include more and more environments, you might detect fewer SNPs. Or, how is the environment coming into this in terms of where you're getting the genetic data from? And have you guys seen anything with the EA as far as the diversity in the betas across these cohorts and how, I guess if you're just using UK Biobank to really expand the sample, that could be an advantage to finding more hits because it's all from a similar environment.
Yeah. So I would not sure I fully agree with a few of the things that you said. So one is I'm not sure, it's not necessarily true that as we expand the sample size we're getting more diverse environments, because initially we were just getting these convenient samples of medical cohorts, and it's not clear that the new cohorts we're adding are any more variable than the old cohorts that we were starting with. It's also not necessarily true that the results that we're getting are the ones that are robust to environmental variation in the sense of varying less across environments than the newer hits we're getting. The way to think about what we're identifying is the average effect of the SNP across the set of environments in our sample. Now then you can look at how much does that effect vary at the cohort level across the cohorts. When the GWAS sample sizes are small, the power of the test to look for that kind of heterogeneity is low. As the sample sizes get bigger, we can more reliably detect that there is some heterogeneity. Like if we look at the EA3 results and with almost 800,000 people, the top hits do seem to show some heterogeneity across the cohorts, and I think that's probably going to be generally true. I think. Yeah, so we may say more about this when the gene-environment interaction lecture and the simulations and applications. Maybe we'll go into some more detail on heterogeneity across cohorts. Okay. So by now there have been a number of large-scale GWAS. A lot of these have come up already in discussions so far. So let me jump ahead to one thing I want to say is just to emphasize the point from earlier. Candidate gene studies were problematic in practice, and by the way, we don't really know that all those results in candidate gene studies were false. In fact, they probably weren't. Probably dopamine-related genes are related to risk-taking behavior and IQ. It's just that it's hard to know, and probably we were getting very exaggerated effect sizes, and so it's just hard to disentangle what's true from what isn't true. And now we're getting robust and replicable results in some of the more recent findings. But the basic idea of candidate gene studies was totally fine in principle, and there were ways you could deal with the various concerns. You could pre-register and use the genome-wide significance threshold in a candidate gene study, or at least some Bonferroni corrected threshold. You could, if you have genome-wide data, you could control for principal components or look within families. You could do them in large samples, and in fact, there have been some very interesting candidate gene studies being done. And for example, we did a study of cognitive ability where we used the SNPs associated with educational attainment as candidates. So instead of getting our candidates from theory of what the biological function was, we took it from established results, and we did a candidate gene study, which we called a proxy-phenotype study, and we found some SNPs that seemed to be associated with cognitive ability at a Bonferroni corrected significance threshold. Okay. So let me go into a little bit of detail on the GWAS of educational attainment. So to continue the story, after my co-authors and I were very disappointed with our candidate gene studies, and we did this test of the candidate gene association with cognitive ability, we came to appreciate that a good way to proceed for social science might be to imitate what the medical geneticists were doing, of forming consortia and getting a very large sample. So David and I, together with Phil Kohlinger, who's at the Vrije University in Amsterdam, founded the Social Science Genetic Association Consortium in 2011. The first problem we faced was deciding which trait to study. And so we did power calculations. We, in retrospect, we overestimated the effect size we should expect. We did calculations assuming an effect size of 0.1% R squared, so 5 times bigger than what we ended up finding as the biggest effect. But based on those power calculations, we use those kinds of power calculations to ask the question of what sort of trait should we study. And in particular, the trade-off was between a trait like educational attainment, which we thought would be available in many medical genetic studies because it was measured as a background variable, but clearly is very, very far from what genes actually do and is highly affected by the environment and probably varies a lot across different genetic effects might vary across environments. So in that sense, it's kind of a noisy phenotype versus a phenotype like cognitive ability or a specific cognitive ability like memory, a specific kind of memory or working memory that might be much more closely related to what genes do and probably have, probably can be measured much more reliably. And we ended up concluding that to do a study of cognitive ability, we might need a sample of like 30,000 people. To do educational attainment, we'd need a sample of like 100,000 to get the same level of power. And it seemed to us like it was going to be much more doable to get the large sample of the noisy phenotype than the still somewhat large sample of the better phenotype. Given that there aren't that many studies that measure cognitive abilities well, it's just not a focus of a lot of these genetic studies, and it's expensive to collect because it takes a long time to administer those tests. So that led us to focus on educational attainment as our proof of concept. So for our first paper, which we finished in 2013, we ultimately ended up getting 41 data sets with a sample size of about 100,000. We studied years of schooling and a binary indicator for college. I'll spare you the details, but it's an enormous effort requiring tons of time by various people. I think Niels really bore the brunt of a lot of Niels Reitveld bore the brunt of a lot of the work in this first paper, where dealing with cohorts who upload results, and then it turns out there were QC problems, as you'll hear about from ISU, very, very common. You have to tell them those results are no good, and then you have to, you don't have access to the data. So you have to figure out, as the meta-analyst, what they might have done wrong based on looking at the meta-analysis results and telling them how to do it differently. And of course, every cohort thinks that they did it right? Like they don't think they made a mistake. So the responsibility is on you to find the mistake. And there's lots of back and forth and trying to organize these people and giving them a deadline. And of course, people are late when you're dealing with 41 cohorts. So it's a huge mess, huge administrative nightmare. But eventually, you get all the results uploaded to a secure central server, and you meta-analyze them. We found 1 genome-wide significant association with years of education and 2 with college. We were then had a replication phase of the project where we got 12 independent data sets with a sample size of about 25,000 people. And we we found that all 3 of the hits replicated at the Bonferroni adjusted 5% level. So this was the paper EA1, published in 2013. It's published in Science. Here's the Manhattan plot for the years of education variable. So you see the 2 significant hits here. The other hit was, sorry, this is for college. Yeah, these are the 2 significant hits for college. The other one was for years of education. Now, after that paper came out, I think it's fair to say that basically nobody believed it. It was, I was saying that, I was saying before the importance of actually doing kind of credibility calculations and not just relying on heuristics. We had done those calculations, and based on them, we thought the results were very likely to be true. But a lot of people looked at the paper and said you only had 16% power based on your effect sizes. So it was a low-powered study. Probably those results aren't true. They were also concerned that we hadn't done enough to deal with population stratification. I think there was also an element of a heuristic that it's social science genetics, so it's probably wrong, which is actually not such a bad heuristic, I think, for applying to results at the time. We then wrote a paper the following year to try to assess how replicable and robust these results really were. We were contacted by 23andMe, who had data on an additional 35,000 people, and they were willing to, they had replicated our results, and they were willing to do some additional analyses to probe more into population stratification as a potential concern. I won't talk about that here. Yeah?
I'm just curious, what do you kind of mean when you say that people didn't really believe it? How did you perceive that? Were you at conferences and people would laugh at you, or it was just I kind of don't know what that means, but I think as people that are interested in this, it seems kind of important.
So our paper, there have been many conferences and where basically our paper was shown, flashed on the screen as the reductio ad absurdum of GWAS, of social science genetics. This is what happens if you take it, you take this kind of GWAS thing seriously and apply it to social science. You end up with no hits, 3, and tiny, tiny, tiny effect sizes that can't possibly mean anything for anything that people might care about. So yeah, it's all this effort for basically nothing. And why would this be published in Science? It's such a it's totally uninteresting. And then there were the medical genetics folks who also just thought it was probably wouldn't replicate. So yeah, we can, if you're interested, we can send you, blog posts from the time and show you, the kinds of reactions. They were almost universally negative.
And even today, I'll mention people that I do work on educational attainment, a GWAS of that, and not even having heard anything about it. It's like, oh, I don't believe that. It's like,
Yeah, that happens.
Part of the reason is historical. If you go back, and I saw somebody do this in a paper recently. You can do it decade by decade, and every 2 or 3 years you'll find a quote in some journal by a human geneticist saying, we're at the verge of a breakthrough in human genetics, and we're about to find all these genes associated with behavioral traits. And until very recently, those claims just didn't materialize. There were new claims being made, and then subsequently they just evaporated when people tried to reproduce the findings. So if you sort of relied on this, if you knew a thing or two about history, there was a good reason to be cautious. People have made similar claims in the past, and they hadn't panned out. So.
I should also say that in historical context, this approach of focusing on this noisy, distal from the effects of genes, environmentally heterogeneous phenotype like educational attainment ran very much against the grain of what was considered to be the right way to do genetics of social science. In fact, I told you we went through these power calculations that led us to focus on educational attainment. We were repeatedly told by various important researchers, funding agencies, that was a big mistake. There's no way, you should not be focusing on educational attainment. You should be studying the proximal phenotypes that are much more closely related to what genes do. And we said, but then we can't get a very large sample for that. And they would say, well, that's what you have to do. Tough, yeah, that's the world. They would all, the other issue was that the thinking was you needed to have a rich set of social science controls, like you need to be able to deal with all the complications that we know, the various things that matter for education. You're going to do a dumb GWAS where you're controlling for sex and age and some principal components. Like there's no way you're going to find anything that's reliable from that kind of dumb research strategy. Okay.
But something worked
Yeah.
out because you got
What?
into science.
Yes. Well, we got into
That doesn't
science.
mean it's true in and of itself.
Yeah, and this was after, the review process of science was not exactly easy. We got, many of the editorial, we got not just the regular referee reports, but also reports from editorial board members of science. It was a major effort to convince them that this was real.
Why publish it in Science rather than some social science journal?
Well, I don't think a social science journal, I would have been interested because it's not clear what the direct relevance is of these results for social science. But I also think it is, if the results are true, it's potentially a very broad interest across sciences. Also science is higher impact. So if we have a chance at science, we certainly wanted to try that. Okay, so we wrote this 2014 paper. This is a summary of the results actually across the discovery sample from the 2013 paper. So the dark bar is from our 100,000 person discovery sample. The dark gray bars are from the replication sample in the original 2013 paper. And these light gray bars are from the 23andMe replication sample. So I've shown here for the 3 SNPs that were significant in the original study, the results in those 3 samples with 95% confidence intervals. And the y-axis is the effect on number of years of education. So what you see is that we're rejecting the null hypothesis in all cases, so the confidence interval never includes 0, and the effect sizes are roughly comparable. So the bottom line of this paper was these results seem quite replicable, and I'm not telling you, the 23andMe results also had more stringent controls for population stratification. Okay, but the results also underscored just how small these effects were, which we argued was actually a major point of the paper. It was smaller than we expected, and I think certainly smaller than was being implicitly assumed in all of the other work in social science genetics, an order of magnitude smaller than what was being seen in the GWAS hits for the medical traits, and it reinforced all of these lessons of needing stringent significance thresholds, very large samples, and so that was a message we played up. Even though our critics pointed to this as saying that therefore it doesn't matter. Okay, so then in 2016, we did EA2. So this was we expanded the sample size to 63 data sets with about 300,000 people. Similar analysis plan to as in EA1, except we were focusing exclusively on years of education. And the bottom line is we found 74 lead SNPs. After we had submitted the paper, the first release of the UK Biobank became available with 110,000 people, and so we were able to use that as a replication, which we included in our resubmission of the paper. So here's the Manhattan plot from the discovery sample. There were 74 orange dots corresponding to the lead SNPs. We've marked here with X's the lead SNPs from the 2013 paper, so they all continued to remain genome-wide significant in the larger sample. This shows the comparison of the effect sizes from the meta-analysis with the UK Biobank replication. So we've shown here on the x-axis is the 74 lead SNPs. The black bars are the meta-analysis results with 95% confidence intervals, and right next to them the UK Biobank replications. The y-axis is the regression coefficient in years of schooling. And so you can see visually that this is a pretty good correspondence between the discovery sample and the replication sample. We can also do calculations of what kind of replication record you would expect given if the original effect sizes we estimated were the true effect sizes. You wouldn't expect perfect replication because there's sampling variation, and we have 110,000 people in the replication sample, so we can calculate how much replication you would expect. And what we found was that the replication record was actually a little better than what our calculations indicated we should have expected. We found that 72 out of the 74 SNPs had a consistent sign in the replication. 52 of them were significant at the 5% level in the replication sample, and 7 of them reached genome-wide significance. So we view that as pretty strong replicability. Yeah.
I don't know if you agree with this or not. I actually find the consistency of the sign to be a more impressive achievement than like the number of 5% significance level. I mean, you know what I mean? Like in a lot of other cases, you see a lot less sign consistency. And some people even claim support for a result when they get the opposite sign. But it's significant. So.
Yeah, I mean, none of these are really perfect indicators of replicability. They're just different ways of assessing it, and you can assign p-values to each of these kind of sets of results. I think I kind of interpreted kind of as a package, that our replication record along any of these metrics was better than you would at least as good as you would expect if they were true.
What do you think? Oh, you go.
Yeah. Yeah.
Can you talk a little bit more about what it really means, replication? So you found your 7 SNPs in one set, then you go and you just test for those 7 SNPs, because it seems like then you can do, in a sense, let's say you actually have whatever, we have 67 samples in one, now you're adding 1 sample. You could basically start playing with doing meta-analysis by reshuffling samples and see, for example, what happens to the distribution of the, right? Or even testing like if the values really are identical or not across different meta-analysis and subsamples. Because like even if you just look at the bars, indeed, the standard errors seem to overlap. But
yeah.
When you have 500,000 observations in one sample, in the other one, a difference of 0.001 may be statistically significant.
Yeah,
right?
no, there's all sorts of different things you could do, and I mean, I think, I think it would be interesting to do other kinds of replicability analyses. What was particularly clean about this setup for us is that we actually didn't have available this other data set when we originally submitted the results. So, there was no kind of fooling around that we could have done. We were just looking in the new data that became available how the results held up. But I agree, there's a lot more you could dig into. Okay, Hui.
Is it possible that in the replication of data sets and the original data sets have overlap information that it comes out?
Absolutely. If there were some of the same people in the replication as in the discovery sample, that would be potentially a major problem for claims about replicability. We think it's unlikely based on, at least it's unlikely to be a problem for a large fraction of the data. I mean, it could be that some of the UK samples that were included in the discovery sample also had participants in the UK Biobank. So it could have affected a very small fraction of the population. We can't rule that out because we don't know who the individuals are. We don't think it's unlikely to be a major source of bias, but it's always something that you should worry about when and not just with replication, also in a discovery analysis, if you have the same person in multiple cohorts, it's going to create a falsely strong signal.
Yeah, I think we can say even more now because in discovery we included some Scottish cohorts and some British cohorts. And at least for the Scots, I think we now know that a couple of those are in the UK Biobank. It's very hard for me to see how that would how that could change our conclusions in any major way. But with hindsight, we probably should have just eliminated those altogether from the discovery sample for the purposes of this exercise.
Yeah, Jose.
So how the findings from GWAS like these inform the creation of polygenic scores?
So that's going to be the topic of Monday. So given the time, let me just defer that until later in the course. Yeah.
How do you explain the dissonance between educational attainment 1 and then the replication in the biobank? Like it seems like you have a very similar sample size, but you have like very different results. Like if that was EA1.
Yeah, that's an astute observation. It's very interesting that we're seeing so much, 7 significant hits in the UK Biobank sample of the same size as our discovery sample of EA1 when we only had one hit for years of education. Exactly. I think that is because we have a more homogeneous environment and a more homogeneous, genotypic background in the UK Biobank sample than we had when we were meta-analyzing the cohorts from EA1. In fact, we're now seeing it, in EA3, we now have enough power to start looking at heterogeneity across cohorts in more interesting ways. And I think we'll get to that, we'll talk about that next week. Yeah?
Given the critiques in the EA2 about possible overlap between discovery and replication sample, are you able to address that in EA3?
I guess we could do an analysis where we just excluded anyone from the UK in the discovery sample of EA3. I think that would have a trivial effect on our sample size if we so we could do that. I don't know if we have done that.
There are also several methods to analyze the sample overlap between cohorts. And so in EA3, for example, even if in cases where there seems to be some overlap, it's very, very, very small. So it doesn't seem like something to really worry about.
Yeah, no, that's right. Also, something else that we're going to be talking about, I think, tomorrow is LD score regression, which is a method that allows you to correct for any sample overlap that might be there in your results. And our results are that I'm quoting you are actually after doing that kind of correction. Okay. Any other questions? Okay. Actually, one more thing to say about the effect sizes is I've been highlighting this 0.02% R² number, but I want to point out that if you look at the effect size in terms of years of schooling, and it's like 0.1 years of schooling per minor allele, that's like a month of schooling, that doesn't sound small at all. I mean, if you compare it to standard kind of interventions designed to affect educational outcomes, that's a pretty big effect. And if you think about, especially about the fact that that's a single allele in a single SNP corresponding to a month of school, you could argue these are not tiny effects. And that's actually there's a kind of general phenomenon that comes up with describing effect sizes in terms of percent of variance explained versus a regression coefficient. They often seem bigger in terms of regression coefficients than an R² because the R² reflects the fact that there are so many other factors that are varying, that are affecting years of education. And so, this particular factor is not explaining much of the variance, even though the allele is corresponding to a moderately sized regression coefficient. It's the R² that matters for power, as you showed on problem set 1. So in terms of the lesson for the kind of sample sizes you need, this means you need huge samples. But some of the criticism that the effect sizes are small might be a little unfair. Yeah?
I was quite surprised that in this graph that all the regression coefficients are positive. Is it because
Oh, yeah, these are aligned so that in the discovery sample they're all positive, because it's arbitrary which allele you use as the reference allele. So this just made it much easier to look at the graph.
This is a 70 of 72. Those are the two opposite sign ones. Or?
Yeah, these are the two opposite signed ones. Yeah, and notice they. Yeah, go ahead.
Say you had a sample where you could control for things like parents' education.
Yeah.
How do you think that would change the results of the GWAS?
Well, I think it would substantially reduce our the effect sizes and the power to detect these effects. I think that would be you could argue that it's over-controlling if your goal is to estimate the causal effects of these SNPs, because the parents' education is partly the result of the parents having the same SNP as the child, and the causal effect of that SNP on the parents, in the parents' education. We've done this with the polygenic score for EA3. And Robbie, what is the result when you control for parents' education on the polygenic score? Okay, so Robbie will look that up. Yeah.
So can I think about the effect sizes, the regression coefficient effect sizes, as like you can aggregate those in the same way that you aggregate the variance effect sizes? So if one of the alleles accounts for a small amount of variance, then I could just add the amount of variance that each one of the alleles accounts for, and that's like the total variance explained.
If they're uncorrelated with each other, which they are approximately, because here.
So
yes.
If each one of these explains like a month of education, and you have 74, a bunch of educational attainment, and you have 74.
No, you can add up the variances explained. For the coefficients, you have to be careful.
That's what I'm asking.
So is it just the square root of 74 then?
No, because the issue is that each of these is a coin flip of which allele you get. So if you want to look at the amount of variation of the population, you need to think about, almost nobody's going to have, 2 copies of the education increasing allele for all 74 of these. Most people are going to, what's that?
Nobody.
Nobody. Yeah.
We could run a simulation and for billions of years it would never happen.
At least pre-CRISPR.
Yeah. Yeah. So most people are going to have close to 50-50, some 50% plus or minus, a little bit of the. So if you, we did this at some point. We kind of looked at what the standard deviation is in years of education that would be due to these 74 SNPs. I don't remember what it was off the top of my head, but that's certainly a calculation you can do.
But just so I'm 100% clear about this, if I was doing the exercise of comparing a person with none of the minor SNPs, none of the minor alleles on any of these SNPs, to the person that has all of them,
yeah,
I would expect
you could just sum the betas in that case.
Yeah.
But it's just a weird experiment because it's a really low probability to low, right? But you're right. But that is the right.
But I would expect that person to have 74 months more of education.
Well, times 2 because this is per allele.
Right. Okay.
There's also
I wanna
prediction too.
understand.
There's no common score.
I want to understand those probabilities a bit, because you talked about it, but I don't fully understand. So what's the probability that you have 2, 3, 4? How does it work? Is it 50-50?
No, it depends on the minor allele frequency. So most of these have close to 50% minor allele frequency, but say 40 to 60% range. So let's say it's a 40% minor allele frequency, and let's say this is the effect of the minor allele. Then there's a 0.6 times 0.6 chance that you have.
To the power of 74, sort of? Comes like that? Is it like 0.5 to the, no. What is the
Well, it'll be some multinomial,
Yeah.
distribution.
Although if we're saying that all of them are
Yeah, if they're all
0, 0,
0.5.
then it's easy, right?
What's that?
If you're saying what's the probability that all of them are the bad state,
Yeah.
then you can just do it to the power of them.
Yeah. Yeah.
Does there just seem to be a relationship between the allele frequency? Sorry, like in the population, like the percent minor allele and also like the effect size?
Yes. Well.
It's hard. So. Yeah. There's a confound with doing a direct kind of comparison because.
Yeah, but clean studies of it do find a relationship.
Yeah. There's a recent paper on height that was able to detect very rare the effects of very rare alleles reliably, and they did have, on average, larger effects than the common SNPs that have been found.
In terms of regression
Effects in terms of regression
Does
coefficients.
that seem true between like 50% and 25% as well, or it's not so clear?
I don't know.
Squirrely, that's fine.
Yeah.
There's not a really strong relationship in the common range. I think you have to get in kind of the more rare range before you're really seeing that relationship take off. But I mean, I think there is a slight one on average, and it's phenotype specific too, right? So.
Yeah. So there's an evolutionary argument for why you should see such a relationship for a trait that matters for fitness, which is that a trait that matters for fitness should go to, if it's a positive. If it's a good allele, it should go to 100% in the population. If it's a negative allele, it should go to 0%. So it should be rare if it has a substantial effect on fitness. For traits that are neutral with respect to fitness, then you might not expect any relationship because it's just going to move around from drift. Okay, so let me just mention EA3. We're currently at 770,000 people. We're going to be incorporating the new UK Biobank data when it comes out in a couple weeks, so that sample size will increase to over 1 million. We're using essentially the same analysis plan as EA2. Here's what we're seeing so far with 770,000 individuals, or at least I think we have more than that now, but as the time that we calculated this plot, that was 770,000. We were finding 529 lead SNPs. And so, I think we're seeing the same kind of patterns replaying itself now with social science that we were seeing in medical genetics, with bigger sample sizes leading to a lot more discoveries and much more robust results. And I'm optimistic that this can continue as the sample sizes continue to get larger. And of course, there's other things we can do with the results besides look at individual SNPs, like create polygenic scores, and that also get better and better as the sample sizes get bigger, as we'll be talking about. Okay, so what are the takeaways? First, be careful which gene discovery research you rely on. Hopefully, you now have the tools to look at a paper and assess, is the sample size big enough? Are these effect sizes plausible? After you hear Patrick, you'll be better at judging whether they've done a good job controlling for population stratification. And those are the things you should be looking for in judging whether these are results that you should follow up on in your own research. Major takeaway message is that individual loci have very small effects on the kinds of behavioral traits we're interested in, at least in terms of R squared. That's something that we argued should really be added to our list of laws of behavior genetics. So David had mentioned Turkheimer's 3 laws of behavior genetics. In a 2015 paper, we argued there should be a 4th law on this list, which is that a typical human behavioral trait is associated with very many genetic variants, each of which accounts for a very small percentage of the behavioral variability. And that having that in mind is really going to help organize a lot of what we're seeing in social science genetics with inconsistent replication records of the small sample traditional candidate gene studies, the fact that with large enough samples we're finding lots of loci, and that that they add up to substantial, that adding up over many of these loci can add up to substantial heritability. Another lesson that's from this in terms of your own research, if you're interested in using genetic data to do social science research, then the best shot you have is to do one of 2 things. It's probably not to look at things like the strongest SNP association with educational attainment with like 0.02% R squared, because unless you have an enormous sample, you're not going to be able to detect that effect reliably. You should either be focusing on polygenic scores that aggregate over many SNPs and can have much larger R squareds. That'll be on Monday. Or you should focus on genetic variants that have relatively large effects, which are not going to be for phenotypes like educational attainment or depression or fertility. They're going to be the kinds of phenotypes we talked about earlier from the medical genetics literature, like the Mr. Big effect on smoking. That's the name for the SNP with the biggest effect on smoking, with like its 0.4% R squared, or 0.5% R squared. That's detectable with reasonable power in a sample the size of the health and retirement study with like 10,000 people. So you could study the effect of that SNP. Ideally, even you'd meta-analyze several data sets to have really good power if you want to look at an interaction effect, say, or ApoE effect on Alzheimer's. Yeah.
Is there like a good, easy tool to calculate power, sample size, kind of stuff, besides G-power? Do you know what I mean?
Well, so one tool to calculate power is the program or spreadsheet that you created on problem set 1. Which the purpose of which was that you would then have a tool that you're very familiar with to be able to bring to your own research. There are also, Sean Purcell has a power calculator online. If you Google Sean Purcell, I'll spell his name. If you Google Sean Purcell power calculator, he has one which is useful for a number of different kinds of genetics designs.
There's also Ronald's method.
Oh yeah, Ronald de Vlaming. Let me write his name too. Ronald de Vlaming has a calculator. What is his? What kind of power calculations can you do with his?
So it also takes into account the heritability per cohort or the genetic correlations between your discovery cohorts.
Yeah. Yeah. And the final takeaway is, you should do these calculations in your own work. Don't rely on our intuitions, which especially in this context can be really off base, and don't rely on simple heuristics that can also go wrong. It's actually not that hard to do these power calculations and these credibility calculations. You're doing them on the problem sets, and it's a good general practice to get into with all of your research, whether it's genetics related or not. Okay, so let's stop there. I'm sorry I took all of Patrick's time from before lunch, but Patrick will start on his lectures after lunch. And so we'll continue at 2.